Hello and welcome back to educator.com. Today's lecture is going to be on DNA replication. As an overview, we're going to talk about the eukaryotic cell cycle once again, and we are going to focus on S phase because that's where DNA synthesis occurs. We'll talk about our major players, so that will be how we recognize the origins. We'll talk about how strand separation occurs, removing supercoils, as well as why we need to remove supercoils. We will then go over the big part of the actual DNA synthesis, and finally, we will go over the final processing, and then we will talk about telomeres and the end replication problem. So, the eukaryotic cell cycle, once again. Okay. So, G1, remember that's just our growth phase. That's going to be about 9 to 11 hours long. S phase, which is what we're going to focus on in this unit, that's where DNA replication occurs, right? That usually takes about 8 to 12 hours. G2 phase, another growth phase, is about 2 to 4 hours. And then mitosis is about 1 hour. And so the normal human cell will divide on average about every 24 hours. And G1 is going to be the most variable timing of that stage. But today, as I said, we're talking about DNA replication. So we're going to focus on S phase. Now, we can't talk about DNA replication without talking about Watson and Crick. Now, remember back from the second unit that we talked about Watson and Crick as being the gentlemen who discovered the structure of DNA. So they published a one-page paper to the Nature of Journal in 1953, and in their journal they said that, hey, there is this molecule, DNA, that we all know about, but it actually has specific base pairing that suggests a possible copying mechanism. Now, this is important because at the time, remember, protein was thought of as being the genetic material because DNA, they didn't see any type of copying mechanism because at the time, DNA looked as a tetranucleotide with the bases coming off on the ends, the phosphate backbone being in the middle, right? So this would be the phosphate backbone. And this would be a planar structure. So you'd have a tetranucleotide here, then so on and so forth. And it just didn't have a way that you could replicate DNA. So Watson and Crick's paper, using Rosalind Franklin's data without her permission, was what allowed everyone to start thinking about this differently. Because remember, now what DNA looks like is this, with the phosphate backbone on the outside and the DNA base pairs in the middle. All right. So to talk about DNA replication, we have to start somewhere. So why not start with initiation? Well, initiation or replication requires DNTPs. Now, DNTPs are deoxynucleoside triphosphate. So this just means it's your, DATPs, DTTP, DCTP, and DGTP. Right? And what it says is that, what this slide says is that not only do we have to have those nucleotides, but we have to have a primer template junction. What exactly is a primer template junction? So let me draw this for you. So if we do DNA, and let's say that's our 3', okay, 5', this is DNA. Let's say that we have this is our template, right? And then here we have our primer, and we'll draw it like this. Now this is going to be the 5' of our RNA. This would be the 3', okay, and it has an OH. So that's our 3'. And what we need is this primer template junction so that the incoming nucleotide can join at this OH. Okay, so what really happens is that we have the 3' OH is attacking the incoming triphosphate, the incoming DNTP. So what it does is it attacks the triphosphate, causing a pyrophosphate, which is 2 phosphates, to leave. And then that pyrophosphate gets cleaved again by an enzyme called pyrophosphatase. Well, the reason I'm telling you all of this is that when you are building molecules, when you're doing anabolic reactions, it needs a lot of energy. Okay? So we have these GTPs, ATPs, TTPs, CTPs coming in. Okay, so in the triphosphate form. But they're actually only adding into the new molecule in the monophosphate form because these 2 phosphates, the pyrophosphate group, is being cleaved off. And when pyrophosphatase breaks this pyrophosphate into individual phosphates, that gives you a high amount of energy. So it has a very large negative delta G reaction, and the KEQ is very high, meaning it's basically irreversible. It's an irreversible, high energy giving to the system reaction. All right, so this is important. And so the DNTP, let's say this GTP, would come in there, and eventually what it would be added as is a DGMP, okay, because the pyrophosphate's been removed. All right, so that's how we need to start with, right? We need that primer template junction. Now, let's even rewind further than that because we need to start somewhere. All right. Well, to get this primer template junction, we need to be able to find where we need to start replicating. So that is where we do origin recognition. And that's done via the origin recognition complex, which you can just call ORC. And that's composed of many different proteins. So the ORC protein, CDC6 and CDT1, amongst others, but these are the main ones. And this is a complex of proteins that recognize the proper DNA sequence for initiation of replication. And it will also recruit DNA helicases to open up the DNA. Now, this is a little separate from prokaryotic replication. So what I've told you right now is eukaryotic. Prokaryotic replication will begin at a single unique nucleotide sequence called the ORI site, or the origin of replication. And what we have happening there is DNA A will bind AT-rich sequences. Do you remember why AT? If we think back, AT has 2 hydrogen bonds. GC has 3 hydrogen bonds. So AT-rich, meaning there are less hydrogen bonds to melt, so it doesn't cost as much energy. So DNA A will bind to those AT-rich sequences at the ORI and start to melt the DNA, and then the helicase can come in and bind. So this is prokaryotic. All right, so how I'm running this whole unit right here is that we'll talk about the eukaryotic, and then underneath in the bullet points, I'll give you the prokaryotic homologous proteins. So here's the initiation of replication. So once again, we have our initiation occurring where we're going to start having something bind at these sites of replication, right? The origins of replication. Now, it's important to know that our bacteria, right, our prokaryotes, usually have a circular genome. So our circular genome is only going to have one site, right? One ORI site. So that's our ORI. And replication is going to occur in both directions until it ends at a termination site down here. Well, our eukaryotic organisms are going to have many different sites, many different origins of replication throughout the chromosome. Okay? And so this is you know, you could have anywhere between a separation of maybe 30 kilobases in between these origins of replication. So based on individual organisms and individual chromosomes, that can still give you anywhere from 10 to thousands of origins on a single chromosome. So this is U, and this is prokaryotes, and this will be one origin. Okay, so what we have is many origins of replication. So we can start, and we'll just zoom in, and what happens is that we have the melting of DNA Okay, so this is all still hydrogen bound. And this, in the middle right here, has started to melt. Okay, so the DNA has melted, and we're going to start loading on our helicases. Okay, and our helicases are actually hexamers, so 6 different subunits of proteins added on, and they'll move in a certain direction. And actually, they will have one at each site, okay, and they look like donuts, and they'll be moving in opposite directions. Okay, once again, we always label our polarity of our strands. So, our helicases, as I've already alluded to, in eukaryotes, those are called MCM proteins, or mini chromosome maintenance complex. And what those are, it's a hexamer, a homohexamer, and it is, we have 2, 3, 4, 5, 6, 7 are the subunits, okay? And they catalyze the separation of that double-stranded DNA helix. Now, what's important is that they, as I said, they bind as a double hexamer, and they will bind DNA after being loaded onto it in G1 phase. This is important. So it's already there. It's already there once we move into S phase, but they're not active. They have to be activated by certain kinases, which is called CDK, which is cyclin-dependent kinase. Cyclin. And DDK, which is DBF4 dependent kinase. And once those are activated, once we get to S phase, then it will start opening up the helix. So remember, G1 gap phase, we're loading the helicases, but they're not active. Once they get activated in S phase, then we can go through DNA replication. Now, we have our helicases binding DNA, and when they are actually active, they will spin the DNA apart, okay, and they'll unwind it, and they do this by using ATP. So for every 2 bases that it pulls apart, it uses 1 ATP molecule, so it's very energy intensive. And this helicase is spinning at about 10,000 RPMs, so 10,000 rotations per minute that this helicase is spinning. So that's very quickly. And this is because we need replication to occur. Remember, synthesis phase, we have 8 to 12 hours, but we also have to replicate over 3 billion bases. All right, so if we're looking at the prokaryotic side of this, we have DNA B in prokaryotes being the helicase, and this is requiring DNA C to bind the DNA. Now, once we have separated some DNA, we're going to have some double-stranded parts of DNA, right? And then we have some single-stranded parts of DNA. Well, these single-stranded parts of DNA need to be kept apart, all right? And so we have proteins called single-stranded binding proteins, and in eukaryotes, those are RPA, okay? Now, RPA, it's just replication protein A. Now, RPA, those proteins are going to bind these single strands, and what it does is it not only keeps them from reannealing and rehydrogen bonding back to their complementary strands, but it protects them from any nuclease activity. Remember, nucleases will cut DNA, and it usually does so, some of them do so non-specifically. So there's nothing to keep a nuclease from coming in and cutting this DNA. And if it were to cut it, let's say at 2 different spots, we can lose a piece of this DNA, right? And that can just go off in space. So these single-stranded binding proteins bind onto that DNA, not only keeping the 2 strands apart, but preventing it from being broken apart by our nucleases. All right. So the protein that does this in eukaryotes is RPA. The protein that does this in prokaryotes is called SSB. So single-stranded binding protein. Now, as I mentioned in our previous unit, that when the helicase unwinds, right, we need to think of our, back to our old telephone cord, right, the coil. When you pull from the middle, you create a bubble. In this case, that's our replication bubble. As you pull apart that middle, on either side of the bubble, okay, so we'll draw it out. On either side of this bubble, we are increasing the supercoiling. So that's just increasing the supercoiling. You're getting positive supercoils ahead of the fork, and you get negative supercoils behind the replication fork. All right? And so you're still going to have supercoils here. Now, if we don't relieve the stress of these extra supercoils, right, we're going to end up breaking our DNA. So that's where our topoisomerases come back in. Now, topoisomerases are proteins that help relieve that torsional stress that's exerted on the DNA from the overwinding. So we have type 1 topoisomerases that nick one strand of the double helix, and we have type 2 topos, or topo 2, that make breaks in both strands. And these are both occurring ahead of the DNA helix, ahead of the DNA helicase, okay, in the direction that the helicase is moving. Now, in bacteria or prokaryotes, it's not topo 1, topo 2 that we call it. The protein that does this is DNA gyrase. All right. Now, remember, MCM, these proteins of the helicase, they get loaded on in G1, they get activated in S, they start unwinding, the single-stranded binding proteins will come in, right, RPA, then topoisomerase 1 and 2 will start cleaving, okay, so it'll nick, okay, whether it be a single strand if it's topo 1, or double strands if it's topo 2, relieving torsional stress. So what we have so far is that helicase is now in there. Okay, let's say that helicase, we're just going to draw it as a donut. Okay. Remember, these are still hydrogen bound, hydrogen bound, hydrogen bound, hydrogen bound, and we will call it okay. So here's our helicase. Remember, we have our single-stranded binding proteins are going to be bound until we need other stuff coming in, other machinery. Topos are nicking. Topos are nicking, okay, maybe double nick, okay, relieving torsional stress. And then we can finally have our replicative polymerases coming in to start DNA replication. So now that we set the scene for the story, let's talk about actual replication. So we have the synthesis of the RNA primer by DNA polymerase alpha. Now DNA polymerase alpha has two different functions. Now one is the primase function, and that primase is an RNA polymerase that will synthesize an RNA primer de novo. Now de novo is just a Latin phrase meaning from the beginning, and that just means we're making it from scratch. So we don't need any previous 3' hydroxyl to add on to. We can just add it from scratch. All right, so then we have the second part of the DNA polymerase alpha, which is the DNA polymerase part of that, and that is where we actually have the synthesis of DNA nucleotides instead of the RNA nucleotides. In prokaryotes, we have the primase function being catalyzed by DNA G. So what this is going to look like is if we have our DNA, for example, we have we'll call this 3', and we'll call that 5'. Now, our DNA polymerase alpha, as well as our other replicative polymerases that we're going to talk about, always synthesize in a single direction, so meaning they have polarity. That direction that they synthesize in is 5' to 3', meaning they always add the new incoming base to the 3' hydroxyl of the previous base. So DNA alpha comes in and it can lay down its RNA primer. Okay? That's about 10 to 20 base pairs. Okay? And it's happening in a 5' to 3' direction. Okay? Then the DNA polymerase part of our polymerase alpha will start making DNA. Okay, and it might do about 20 base pairs. Okay, leaving us with a 3' that has a free OH. Now, this is how, once again, if we look back, this is our primer template junction now. Okay, we have DNA, and now we can bring in our replicative polymerases to continue on to that. So, why do we even need DNA polymerase alpha? Well, the reason being, pol delta and pol epsilon are replicative polymerases. First of all, can't synthesize de novo. Okay, so they wouldn't be able to lay down this first base, right? And they can't add to an RNA. So they need not only the RNA part, right, so that we have the primase function laying down the primer, but we need this 3' hydroxyl that is a part of the DNA base. Okay, so now we can have the rest of our polymerization synthesis occurring this way. And we'll talk about that in the next few slides. So once again, this whole sequence right here is polymerized or synthesized by DNA polymerase or pol alpha, right? And this, remember, is the original DNA template strand. Now, remember, always we want to label our polarities of our DNA strands because that's going to be able to tell us in which direction we are going to synthesize DNA because it's going to be in the 5' to 3' direction. So let's go to the leading strand. All right, so the leading strand is synthesized by DNA polymerase epsilon. All right, now DNA polymerase epsilon still has a 5' to 3' directionality, but it has a 3' to 5' exonuclease activity as well, which this we can just think of as an eraser function. Okay, and it is called proofreading. Meaning, if it has added the wrong base, it can back up and erase it and then put in back the correct base. So, DNA polymerase epsilon synthesizes the majority of the leading strand. And processivity of this enzyme is increased by its association with pCNA, which is the sliding clamp, okay, and it's called the proliferating cell nuclear antigen. And it's also, this pCNA is loaded by RFC, which is replication factor C. All right, so what is processivity? Now, processivity, all that means, in layman's terms, is that it increases the amount of bases that can be synthesized with each time it interacts with the DNA. All right, so really what it is, it's the enzyme's ability to catalyze consecutive reactions without releasing its substrate, which in this case is DNA. So the higher the processivity, the more bases can be synthesized before the enzyme falls off and has to then get back onto DNA. Okay, so let's write this out. So we have our replication fork opened up, all right? Okay, the helicase moving in that direction. We will call this the 5', the 3'. Okay, in black is going to be our parental strand. Okay, now remember what we have is we have the primase function and then the DNA polymerase function. Right? This is all done by pol alpha. Okay, and this will be the 5' of that. Okay, now after pol alpha has thrown down about 10 base pairs of primer, about 10 base pairs of DNA, then we get DNA polymerase epsilon coming in, taking over and synthesizing, or let's not use red since we use that for the primer. We have DNA polymerase epsilon coming in and synthesizing and following the helicase. All right, so this is called the leading strand because even though it's following, it is actually the one that is going faster and longer than what's called the lagging strand. Another name for the leading strand is the continuous strand. Now that makes more sense when we talk about this. It's continuous, meaning we don't have any stops. Now, DNA polymerase epsilon, theoretically, could continue synthesizing after being primed a single time all the way to the end of the chromosome. All right. Now, what happens is as the helicase continues to move toward the right of our screen, it will open up more and more DNA. So, if we just erase this, and we open up this DNA, okay, so now we're having the helicase moving even further. Okay, well, as long as the helicase keeps opening up DNA strands, right? Opening up free single-stranded DNA. Well, pol epsilon is just going to keep polymerizing, okay? Keep synthesizing DNA. And this is the really simple strand, the leading strand. So this, once again, in green, as well as the primer associated with it, is the leading strand. This black part is the parental strand, and this is the leading strand template. And we're moving in this direction. So this would leave us with our 3' right here. All right. So that's the more simple of the two strands. So now let's talk about the lagging strand, or the discontinuous strand. I already drew out what I had wanted to show on this slide. But one more time, as the helicase moves, okay, translocates from this area to this area, we are going to, okay, we are going to open up more and more, allowing the proper synthesis of the leading strand. Once again, always the polarity. All right, good. So what is our 5' to 3' exonuclease activity? Well, an example of that would be if we have a 5' to 3' sequence of DNA, and let's just say it's A, T, G, C, A, T, G, C. All a 5' to 3' exonuclease activity, remember exo means outsides or the ends, nuclease, okay, it's breaking up nucleic acid, so all it is, it's just an eraser function from this side. Okay, so right there, you just saw 5' to 3' exonuclease activity. All right, so the lagging strand. The lagging strand is a little more complicated than the leading strand. Now, the leading strand, once again, is continuous. The lagging strand is what we call discontinuous. Okay. So, DNA polymerase delta, this is delta, will synthesize the majority of the lagging strand DNA. Now, its processivity is also increased by associating with pCNA, which is loaded by RFC. But what we see different in the lagging strand than we see in the leading strand are what are called Okazaki fragments. Okay. Now, what is synthesizing this in prokaryotes is DNA polymerase 3, okay, not DNA polymerase delta as in eukaryotes. Okay, and if I can draw this out for you, let's go back to our nice little replication bubble. All right. Show you the lagging strand. So here's our helicase. Moving in this direction, we have our polarity. This is our parental, remember. I'll show you our lagging or our leading strand. Remember, this is pol epsilon. This is pol alpha. Okay, so what we have on our leading strand, we're following the helicase. On our lagging strand, we're going the opposite way of our helicase, meaning we're going in this direction. All right, so what we have is, for example, we lay down a primer, okay? We lay down that little piece of DNA, right? All by pole alpha. The 5' is over here, meaning we have to go to the left side. Well, pole delta is what's going to synthesize the majority of this strand. Okay, so this is pole delta. Pole delta is usually only going to synthesize about, let's say, 200 bases at a time before it falls off. Whereas pole epsilon can do thousands or tens of thousands of base pairs at a time before it might fall off. And theoretically, all you need to do is prime, so add a primer to the leading strand one time, and you can synthesize all the way to the end of the chromosome. Whereas that can't happen with the lagging strand. And the reason why has everything to do with the polarity of the polymerases as well as the polarity of the helicase. So as the helicase keeps going to the right, okay, we will free up more and more single-stranded DNA. So look, we've freed up enough single-stranded DNA, we can throw down another primer. Okay, so we have pole alpha throwing down a primer, a little bit of that DNA. Okay, this is its 5' end. And then we have pole delta coming in and going about 200 base pairs or so. All right, well, as the helicase is still there, we can't go any further. We need to wait for more of this single-stranded piece to come away. So if we just say that the helicase keeps going in that direction and is opening up more, let's say it's opened up more. Ooh. Okay, let's say it's opened up more. So our leading strand never has to be reprimed again. It keeps going. And our lagging strand has gotten some opening. Okay, so now we can throw down, let's say, another primer. Okay, go through there. Then we have pole delta. All right, so pole delta. This is pole alpha. And remember our 5'. This would be the 3' end of that, right? The 3' end of that, the 3' end of that. Okay, so it's the discontinuous strand because it has to wait until helicase frees up more single-stranded DNA for a new primer to be thrown down. Now, as you can see, we would have RNA in our DNA complex, right? So we need to process those fragments. And in case I forgot to mention, when we have the parental DNA, (5', 3'), remember, primer. This is our leading strand. This is our lagging strand. Okay, each one of these primer and let's see, each one of these primer DNA pieces on that lagging strand, that is called an Okazaki fragment. Okay, so each one of those, right now I've written it as having 2 different Okazaki fragments. Each one of the primer laid down by pole alpha with a little bit of DNA also by the pole alpha, and the DNA synthesized by pole delta. Each one of those little pieces, those discontinuous fragments, are called Okazaki fragments, right? And we have RNA in each one of those. So we have a lot of RNA there on the lagging strand, and we have just this, hopefully, this one piece of RNA on the leading strand. But we need to get rid of that RNA. We want only DNA in our DNA double helix after it's been replicated. So how do we go throughout doing this? We have to go through Okazaki fragment processing. Now, Okazaki fragment processing happens when we have pole delta, FEN1, and DNA ligase all coming into play. Now, if we're talking about happening in prokaryotes, this is carried out by DNA polymerase 1 and DNA ligase. And in E. coli, replication termination actually occurs when we have this TUS protein binding to a TER site on the DNA. So it's a little different. That's why we're focusing on eukaryotes. So when we have this Okazaki fragment processing, what we end up having is, as pole delta, if we draw it out again, okay, remember, this was done by pole epsilon. This was done by pole alpha. This is your leading strand, right? From here to here, that's your leading strand. This is your leading strand template. This is your leading strand. Okay, up here we have your lagging strand template. This is helicase. Always moving in a 5' to 3' direction. And over here, we have, that's a 5'. It's a 5'. Okay. Now our Okazaki fragments. Okay, each of these okay. Now, for processing, what actually occurs is that we have polymerase delta actually displacing or going underneath. Okay, if we take the arrowhead off this one, it actually remember, this is all going to be hydrogen bonded. Hydrogen bond, all this stuff, right? Okay. Now, pole delta, as it's polymerizing in Okazaki fragment, it's going to come under and displace about 1 to 2 base pairs, okay, 1 to 2 nucleotides of this flap, okay, of this primer. So what it looks like up close is normally This is your DNA. This is your primer. And when the polymerase delta comes in, it actually ends up lifting up this primer and allowing the pol delta to come in, okay, and continue polymerizing. And then what we have is FEN1, flap endonuclease 1, is a protein that comes in and cuts the non-base paired primer out. So it'll cut that, and then look, we've gotten rid of some. Then what happens again? We have DNA, we have this just continually repeating. So DNA polymerase delta keeps coming in. Keeps coming in. Further and further. Okay, displacing more and more of that RNA. That RNA will pop up again. Okay, FEN1 comes in, cleaves, and it's basically let free. And that continues to go in even through maybe some of the DNA bases. So what we see over here, if we're watching it, even some of the DNA bases, and it's there, and what's happened is we've erased that RNA primer. And that's what we call Okazaki fragment processing, or the removal of Okazaki fragments. And we would have to do this for each one of the primers seen, even the ones on the leading strand, right? Now, once we have that, we just have a nick, okay? So we don't have a nick in the backbone. We just have, we have a nick in the backbone, and that's all that we have. So we have then DNA ligase coming in and sealing that nick by covalently joining that free 5' phosphate with that incoming, with the 3' hydroxyl. So the 5' phosphate, the 3' hydroxyl get sealed together via DNA ligase to seal the nick in the backbone. All right, perfect. Now, an example of our 3' to 5' exonuclease activity, we've talked about it before. Remember, DNA pol epsilon has this. If I were to write out a sequence, A, G, T, C, A, G, T, C, 3', okay? All that is, it's an eraser function, okay, like this. We can erase, 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 erase. And that's your 3' to 5' exonuclease activity. And this is important when, let's say, bound to, if we write this back out, A, G, T, C. Okay. If this were bound, if this is newly synthesized, right, and this is T, C, A, G, T, T, C, A, G, right, this is 3', 5'. Let's say that this is the parental, and this is the nascent or newly synthesized strand. Well, what if it accidentally put in an A right here? Okay, that's the incorrect base pairing. We know this. G's with C's, A's with T's are proper. So it can use its exonuclease activity to erase that and then come back and properly put in the C. So this is important. So, for example 3, I want you to draw a replication fork and label the Okazaki fragments in order of when they were synthesized. All right, so we'll draw this together. We'll do our parental. Helicase, right? Always go in that direction. Remember, do not forget your strands polarity. Okay? So let's first of all do the easy one, the leading strand. Primer, right, with a little bit of DNA. Then the new polymerase comes in, synthesizes all the way behind that helicase. All right, remember this is pol epsilon, pol alpha. Now, over here we have the same thing. Now, if I drew, okay, primer, a little bit of DNA, some synthesis. Primer, a little bit of DNA, DNA synthesis. Primer, little bit of DNA, DNA synthesis, right? All right. Let's not forget. Okay. And remember, we have the red is pol alpha. The blue over here is pol delta. Okay. So what I want you to do is label the order in which these Okazaki fragments were synthesized. So if we think back to the fact that our Okazaki fragments are discontinuous, right? Our lagging strand is discontinuous. The reason is we have polymerization on this top strand, right? Going leftward. But we have the helicase direction going rightward. So we have to wait until the helicase frees up more DNA. So the answer to this question is the most recently synthesized Okazaki fragment is the one closest to the helicase. Okay, and then this one is older and this one is the oldest, right? Okay. Now, so far I've shown everything in simplicity, and actually this replication fork, they often go in both directions. So you would actually have a helicase going in this direction as well, and at the midline, we're just going to say this is the midline because I've already drawn over here. Actually, we'll erase. So at the midline, we actually have a switch of this is the leading strand, right? This is the lead. This is the lagging. All right, we still have this is our 3', this is our 5'. But as the helicase is going in the other direction, right, as of right here, okay, this means we have a switch of the lagging and leading strands. So actually up here, we'll give this nice little delineation of a midline. What we have is this becomes the leading strand following the helicase, and this becomes the lagging strand. Okay, so once again, our 5', 5', this would be a 5' over here. So I've shown it in simplicity so far, but this is really what's happening. The replication fork, when it fires, it's going to go in both directions. So you're going to have over here the top strand being the replication being the lagging strand, the bottom strand being the leading strand. But when we're looking in the other direction, we actually have the top strand being the leading strand and the bottom strand being the lagging strand, the discontinuous strand. So hopefully this was clear enough for you to see how we have the leading and lagging strands. And let's label these, which one was the first or the newest, sorry, the newest or the one made. So this is the newest one made. Okay, number 1 and 2, so newer. Okay, so actually, let me delete that and make it a little easy for you in saying this was actually the first one made, this was the second one made. This was the first one made, this was the second one made. But the one closest to the helicase is the newest. So the Okazaki fragment, closest to the helicase, is the newest. All right. Now, when we've talked about the fact that everything needs to be primed, we've talked about Okazaki fragment processing, but we don't talk about what happens when we get to the very ends of the chromosomes, at the telomeres. All right, so what is a telomere first and foremost, and why does it bring about what's called the end replication problem? Well, telomeres are regions of repetitive nucleotide sequences. In humans, that sequence is TTA-GGG. Okay? And that's found at the end of each chromatid. And if we see down here at the picture, the things lit up in white at the ends of each chromosome, okay, those are our telomere sequences. Okay. Now, our telomeres possess a 3' overhang. The reason being, when we go through and take off that primer, all right, if we look back, here's our DNA, the end of our chromosome. So this is 3', 5', 3', 5'. This, remember, was a primer. So when it gets removed, we have a little bit shorter 5' end on the very ends of our chromosomes. So we have a 3' overhang. Now this is important, and we'll talk about it in the next slide. So telomeres will act as caps for chromosomes, and they do that by binding a bunch of proteins. And these proteins help for a couple different things that I've mentioned before. First of all, they protect the end of the chromosome from exonucleases. So remember, exonucleases could just come in here and start erasing, right? Right? Keep erasing, keep erasing, keep erasing, right? Exonucleases. And if we have a gene here, if this is gene sequence, if we keep erasing with nucleases, we're going to get into a gene and we're going to affect that gene, and that can be big time problems. It can either lead to apoptosis, which is okay, the natural death of the cell, or it can lead to cancerous cells. Okay? Now, other than protecting from exonucleases, now these telomeres, by binding proteins, they protect from fusion with other chromosomes, and fusion of a couple chromosomes together will affect how they get segregated into daughter cells, and whether it be mitosis or meiosis, and this can affect how many chromosomes you get. You can have what's called, for example, let's say trisomy 21, right? That can lead to mental retardation. There are other types of fusions, and we're most likely talking about fusions. Strike that. Take out the trisomy 21 part. That's just an extra chromosome. So when we're talking about fusion with other chromosomes, we can have chromosomes breaking or just binding together via their end regions, their telomeres, and that will make it to where certain cells do not even have, let's say, for example, a chromosome number 20. And then the other chromosome, the 20 that should have been in that cell, is in another cell, so that cell has 2 chromosome 20s per se. A lot of this can cause cancer. Now, telomeres and the end replication problem that I talked about before, the fact that we have to use a primer to initiate DNA synthesis is followed by the removal of that primer, right? We obviously have to take out that primer. We don't want RNA in our DNA. That causes the progressive shortening of the DNA after each replication cycle. So if we draw our telomeres again, so we have the 5 prime, 5 prime, 3 prime, 3 prime. Remember, this was a primer that we've had to remove. So we have a 3 prime overhang. Well, if I go like this, if I'm going to replicate again, right? So after one replication, okay, this one is going to be even shorter, right? Here we go. Because since this was the template, right, we're only able to synthesize, remember, 5 prime to 3 prime, 3 prime to 5 prime. So this ends up getting shorter and shorter, okay? And then we can do that even more. Okay, 3 prime, 5 prime, 3 prime, 5 prime. So this is after it's all been processed, and as we can see, the chromosome used to be to here. Now, we've gotten shorter with each replication. And shorter and shorter. And at which point, we could eventually end up inside of a gene, right? And if we start deleting parts of our genes, as I said before, we're going to either go through apoptosis, or one might end up becoming a cancerous cell. So this causes the progressive shortening, and in most eukaryotes, we have the ends of this linear DNA, so our telomeres, being replicated by a unique mechanism that we call, using the enzyme called telomerase. So telomerase is that protein. So telomerase is that protein that can help alleviate this end replication problem. So it's a ribonucleoprotein. That means it consists of an RNA as well as protein, and it uses this small RNA molecule as a template for reverse transcription. Remember, reverse transcription is making DNA from RNA. It uses complementarity between its RNA template and the telomere DNA sequence, and that binds the DNA, and it will eventually extend the 3' OH of DNA by reverse transcription. Okay, so it'll extend it, then it'll translocate, extend more, translocate, extend more, and then DNA polymerase alpha will come in and synthesize the new DNA using this new longer 3' end as a template. All right? So this is happening here. Okay. We have the binding here. We have the extension here. All right. Now, this is just another way to look at telomerase. Telomerase as a factory. Okay, this is the protein part of it. Okay, here's the RNA part of it. And this is complementary to the telomere sequence of whatever organism, in this case humans. So it binds to that. then we have a synthesis, we have an extension of that 3' end, and eventually we can have a polymerase come in, be primed by that DNA polymerase alpha, and extend 5' to 3' as always. Okay. So one of our, the last example for this unit, I want to mention reverse transcription because I talked about it before. Remember the central dogma: DNA, RNA, protein. DNA to DNA, replication, which we talked about all of today. DNA to RNA, transcription. RNA to protein, translation. Reverse transcriptase, okay, that's an enzyme that is capable of doing reverse transcription. What that does is it turns RNA into DNA, just like telomerase. Some examples of enzymes that have reverse transcriptase capability, probably the most famous one is HIV. Another one is the hepatitis B virus. They both contain reverse transcriptase enzymes, and really, if we talk about any of the viruses in the retrovirus family. Okay. So one of the last things I want to talk to you about, telomerase. We have it usually only being active in germline and stem cells. It's reactivated, however, in many immortalized stem cells, in many immortalized cells. So immortalized cells, that's usually code word for cancer. All right, so our cells are supposed to eventually die. Okay, so as the telomeres shorten, that is somewhat maybe correlated with aging. Right, so telomerase may regulate aging and senescence. Senescence just means the not dividing anymore, so you're basically in G0 phase. And it does that by increasing what's called the Hayflick limit. And the Hayflick limit is the measure of the number of times a normal human cell population will divide before cell division stops and it undergoes senescence. And this is usually regulated by getting to that critical limit of the telomere. So if you increase telomerase in cells, if you increase the activity, you can lengthen the telomeres and decrease the critical lengthening limit, so therefore increasing the Hayflick limit. However, we don't want cells to be able to divide forever. When cells can divide forever, they become cancerous because they often grab more and more mutations. So in the lifetime of a cell, they get mutated over and over again. And as they start to pile up and as they start the mutations get into genes, that causes the cell to go haywire and become cancerous. So we don't want that. So some cells have instead of having telomerase activity, they have a different way of lengthening their telomeres called alternative lengthening of telomeres. And that'll occur in some telomerase negative cells, as well as some tumors. And this is where we have the 3' end of the DNA forming what are called T-loop structures. And they use the process of strand invasion, which we're going to talk about later in lecture 8 or 9 when we talk about homologous recombination. So it uses strand invasion to use one strand of the template, or it uses one strand as a template for new DNA synthesis. So this is important because we can't always use telomerase inhibitors as a way to kill cancer cells, because some cancer cells have found a way around it by using this. So that is something that is always having to be looked into. So I leave you at that, and I would like to invite you back. Thank you for visiting educator.com. Please make sure you come back and see me again.